my slides now?
Yes.
Yep.
Yes.
Okay, perfect. So I'll give a really brief introduction to CAMI, which is the topic of the following talks. So CAMI stands for Critical Assessment of Metagenome Interpretation, and it aims to establish standards in metagenomics or in computational metagenomics by getting the community to talk together, define what's necessary, and organizing benchmarking challenges that people then can use to play or submit results for their tools, and then there's a shared evaluation of those results, and all of this helps to establish where the field is at and where the challenges are. CAMI has been around since 2014 approximately, and that's when it became evident that there's currently a big challenge in computational metagenomics because it's such an active field. I mean, we've heard today again about so many nice methods and different things that are going on, and a problem is if people benchmark on all kinds of different data sets or use different methods or metrics, because then it makes it's really difficult to compare those methods of different papers with each other directly by reading the papers and to kind of understand what problems they're particularly well suited for. And that's why together with Alex and with Thomas Ratti, we founded CAMI in the scope of a metagenomics program running at the Isaac Newton Institute in Cambridge. And we got a lot of really good feedback starting right then, and continue to gather this and organize meetings for people to provide input over the following years. And the aim is really to be able to provide a comprehensive and objective evaluation of computational metagenomics software to help everyone to see what their methods are good for, what they can be used for, and for applied researchers to quickly find what they're looking for. And so CAMI is not the first tool. Actually, there's a lot of these critical assessment challenges. And the most well-known one is CAS, probably, which has been running since 1994. And it's really made a difference to that field of protein structure prediction, where it established where the state is at in this field. And they are also running benchmarking challenges, as are CAFA and CAMDA, and all of these initiatives that have these similar aims. So in CAMI, we also let the community decide, like, what are the principles that CAMI should follow? And so the design decisions are made by the community. How should the data sets look like? What evaluation measures should we use? And how should benchmarking be done? What should the data sets look like that we benchmark on? So obviously it should include unpublished data, because the big advantage of metagenomics is we get data from organisms we can't obtain in pure culture. Thus, until it is the advent of genome binding methods and marks, they were also not represented in public databases. So we have people providing data that's not in public databases yet to kind of simulate that situation, and also provide a range of benchmark data sets that reflect all different scenarios that are common in the field. The evaluation measures should, of course, be informative to evaluate developers, but also meaningful to applied people. So not really complicated and abstract, but kind of easy to grasp and have a value for applied research. And really important is that everything is reproducible. So regarding data generation, the programs that are being evaluated, and also the evaluation process itself. And CAMI focuses on common method types and categories in the field, trying to avoid overlap with other benchmarking initiatives. So we had this first CAMI challenge, which we organized starting from 2014, just to give you a brief timeline. And there was a lot of discussion at different workshops, ISMB, ISMB, and hackathons. And then the challenge ran in 2015. And we have the same structure that we also continue to use, that it starts at some point, then there's a deadline where people that are working on raw reads can submit assemblies. And then after this deadline, assemblies are being provided. And then there's a second deadline where people that are using these assemblies can submit their results. So that, there's this first assembly deadline. And so the results of that challenge, there were public workshops for evaluating the results and so on, and eventually it was published. I'm not going to walk you through the details of the results, but just tell you a bit about basically, so in 2015, we had around 40 people that wanted to participate in the challenge. Not everybody managed to submit on time, so we made the intervals of the challenge a bit larger, so there was now more time for people. By 2020, now when we're starting or running the next challenges, we had over the years more than 350 registrations accumulated. And yeah, here you see some of the people who were participating in the benchmarking workshop back in 2016 in Cambridge. So a lot of, yeah, really nice people working in metagenomics. For instance, Chris Quinns or Alex is here, Todd Triingen, yeah. So one of the things that we also provided is the benchmarking software and the benchmarking and the data simulation methods. And so if you want to benchmark your method, there's now different software packages you can use, which calculates the metrics that are used in CAMI, the visualizations. So there's AMBER, there's OPAL, CAMI text for labeling raw genomes as a gold standard, MetaQuest, which the developers adapted for CAMI, and there's CAMI-SIM for simulating microbial communities. Yeah. And right now, at the moment, the second round of CAMI challenges is running, for which you will be hearing some results. And these again focus on common tasks in the field, so taxonomic profiling, metagenome assembly, now including also long-read data, and genome and taxon binning. And we also had a clinical pathogen detection challenge, which I thought was really cool. So this was a data set, provided really an actual patient case where they tried metagenomics as a way to find a pathogen. And the pathogen was later on confirmed in the lab. And so can the methods in the field find this pathogen? So the data, and again, included unpublished data, larger data sets than before, because that was a wish of the genome binning community. They wanted just many more samples, also long reads, PacBio data, and in the Rhizosphere Challenge, Oxford Nanopore. Now environment specific, so derived from specific 16S profiles, genomes were sampled to simulate those abundance distributions. We had a strain madness challenge. So if you want to know if you can resolve strains, that's your data set. It has a lot of strains. And this pathogen detection challenge. We had for the first challenge around 192 submissions from 36 programs submitted by 21 teams. These are most of the people contributing. The list is growing. We don't have the method developers in there yet. So you see there's a lot of participation all around the globe. And if you want to get involved, there's a website. The Microbiome Cozy site points to CAMI and the CAMI data processing site. You can post what's going on Twitter. You can write us anytime. If you have a method to benchmark, the Rhizosphere Challenge is currently ongoing, long read data. So for all of you who've developed nice methods, maybe there's something to play with. Let us know what you want to do, what kind of challenges would be exciting, or if you have some data to contribute. And yeah, with that, I look forward to the talks that are following now.
Thank you, Alice. That was very informative. We have two questions. Maybe we'll take the one from the anonymous attendee. Does CAMI have real-world data time series WGS data? Are there real WGS time series data in the challenge?
No, right, not. There is a time series, but that's not modeled from a real-world profile. But if this is something interesting to do, we could put it on our list. So there's one data set, 5 sample time series data set from CAMI1, but it's not modeled from real-world abundance distributions.
Excellent. Thank you. And
yeah. Okay.
Perhaps